Fermented foods and the microbes they deliver are quietly reshaping how scientists think about aging, not as a purely cell-intrinsic countdown, but as a dynamic conversation between our genome, the environment, and the trillions of microorganisms that live with us. Kimchi, yogurt, kefir, sauerkraut, natto, tempeh, kombucha, and other fermented staples are more than flavorful additions to the plate. They're sources of live microbes and microbially produced metabolites that can influence the chemical marks that sit on our DNA and histones, the very marks, methylation, acetylation, etc., that control which genes are turned on or off. Emerging evidence, from mechanistic lab work to animal models and early human studies, paints an encouraging picture. Regular exposure to beneficial microbes and their metabolic products can nudge epigenetic programming in directions associated with lower inflammation, improved metabolic resilience, and markers of slower biological aging. Mechanistically, the connections are elegant and plausible. Many probiotic bacteria common in fermented foods either produce or modulate nutrients that feed 1-carbon metabolism, the biochemical engine that supplies methyl groups used in DNA methylation. Folate, certain B vitamins, and other cofactors are either synthesized or made more bioavailable by gut microbes. These feed acetylmethionine (SAM) production, the universal methyl donor. When the microbiome helps maintain healthy SAM/SH, acetylcholine, balance, it supports orderly DNA methylation patterns rather than the chaotic drift that often accompanies aging. Parallel to this, short-chain fatty acids, SCFAs, such as butyrate, produced when fiber is fermented by gut bacteria, act as histone deacetylase, HDAC, inhibitors. That's important because histone acetylation controls chromatin accessibility and gene expression. By modulating HDAC activity, SCFAs can promote expression of genes involved in stress resistance, repair, and anti-inflammatory signaling. In short, microbes and their metabolites provide the substrates and signaling molecules that directly interact with the epigenetic machinery. Beyond these core biochemical links are potent immunometabolic pathways. Fermented foods can help preserve gut barrier integrity and reduce translocation of pro-inflammatory bacterial components like lipopolysaccharide, LPS, into the circulation. Lower systemic LPS and reduced gut-derived inflammation translate into a calmer immune baseline, and chronic low-grade inflammation, inflammaging, is a major driver of epigenetic drift and accelerated biological age. Fermentation-derived metabolites, indoles from tryptophan metabolism, short-chain fatty acids, polyamines such as spermidine produced by some microbes, also act as signaling molecules that modulate immune cell programming and can change the epigenetic landscape of immune and epithelial cells, promoting regulatory, repair-oriented gene expression rather than pro-inflammatory programs. What about evidence in humans? Direct, large-scale trials linking fermented food intake to durable shifts in DNA methylation clocks are still limited, but the existing body of work is promising and growing. Animal and cell studies provide clear mechanistic support. Probiotic strains and microbial metabolites change methylation and histone marks in tissues and can restore youthful patterns in models of metabolic or inflammatory stress. Small human trials and observational cohorts show consistent, complementary signals. Fermented food consumers often display lower systemic inflammation, improved metabolic markers, better insulin sensitivity, more favorable lipid profiles, and healthier gut barrier biomarkers. A handful of targeted human studies report that probiotic supplementation or diet patterns that encourage beneficial fermentation, high-fiber plus fermented foods, can produce modest, favorable changes in methylation at genes related to immune regulation and metabolism, and some pilot interventions indicate that these shifts can translate into modest reductions in calculated epigenetic age. These results are early but promising. They suggest microbial modulation is a feasible lever to influence human epigenetic aging. Importantly, the effects are system-wide rather than narrowly local. Microbe-driven epigenetic changes in gut epithelial cells can have endocrine-like consequences because the gut is an immunometabolic hub. Altered cytokine profiles, circulating metabolite mixtures, and changes in bile acid signaling all carry epigenetic and functional impact to liver, vasculature, brain, and adipose tissue. This explains why fermented food patterns often correlate with improvements in cardiovascular risk markers, cognitive resilience measures, and overall physiological function that align with slower biological aging. From a practical, positive standpoint, the implications are straightforward and achievable. Regularly eating a variety of minimally processed fermented foods, plain yogurt or kefir with live cultures, traditionally fermented kimchi or sauerkraut, tempeh and natto, miso in moderation, and low-sugar kombucha, helps introduce diverse, beneficial microbes and encourages metabolic outputs that support epigenetic health. Pairing fermented foods with prebiotic fibers, vegetables, legumes, whole grains, maximizes SCFA production and helps newly introduced microbes thrive. Because epigenetic effects tend to be cumulative, consistency matters. Habitual inclusion of fermented foods is more likely to produce meaningful shifts than occasional consumption. A balanced tone is important too. Fermented foods are not a magic bullet that will reset, age, or permanently rewrite your epigenome on their own. Individual responses vary with baseline microbiome composition, genetics, diet context, and health status. People who are immunocompromised, severely ill, or on particular medical regimens should consult a clinician before consuming live fermented products or high-dose probiotics. Quality also matters. Industrially processed products often lack live diversity or are laden with sugar and additives. Traditional, low-sugar, minimally heated preparations preserve the microbes and metabolites we want. Looking ahead, the science is ripe for larger human trials that combine precision microbiome profiling, dietary intervention, diverse fermented foods plus fiber, and serial epigenetic clock measurements across tissues. We can expect the field to refine which strains, metabolites, and food pairings most reliably produce beneficial epigenetic remodeling and which subpopulations benefit most. For now, the accumulated mechanistic and early human evidence offers a bright, realistic conclusion. Fermented foods, by supplying live microbes and metabolites that fuel one-carbon metabolism, inhibit harmful epigenetic enzymes, reduce inflammaging, and support gut barrier and immune balance, represent a simple, culturally rich, and low-risk strategy to help nudge the epigenome toward patterns associated with slower systemic aging.